We have sequenced close to 1,000 breast cancer exome, and these data was released last year during the World Cancer Day, and we have released this data in Bharat Cancer Genome Atlas database, and we have analyzed the data, and this data recently published online in BMC Cancer journal. And the importance of these research is we have identified unique changes or unique mutation in Indian population, which is not reported for other populations so far. So one of the major findings from this article, this research, is like in Indian population, we see lot of mutation, lot of inherited mutation in non-BRCA genes. That means that these findings insist for Indian population, we have to go for multi-gene test. Then only we can able to identify what kind of changes happening in a particular patient from Indian origin. And particularly, we have identified a mutation in gene called RECQL. The mutation what we identified, which is more than 3%, which is not reported so far in any other population. And for the same gene, they reported other mutation for other population like a Chinese population, Quebec and Poland population. Though they identified other mutation, but the frequency is less than 1%. Whereas here we identified a new mutation, the frequency is more than 3%. And that gives an indication that we have to go for multi-gene family testing. In addition to that, we see more than 50% of the mutation from non-BRCA inherited genes. As well as we see mutation particularly in the genes that are involved in homologous recombination. As well as we have seen a mutation in a gene called DPYD. This gene basically involved in chemotoxicity with particularly the drug called 5-fluorouracil. When the mutation what we identified in this DPYD gene, it will give a cytotoxin. Basically, when you have this mutation, the patient with this mutation, if you treat them with 5-fluorouracils, you will have cytotoxicity. So that means before you go for the treatment to identify it, because we see the high frequency of this particular mutation. So before we decide for the treatment, better we do the diagnostic test whether you have that particular patient have the mutation in the particular genes, and if that is there, then we have to change the strategy for the treatment. So the bottom line is this study identify unique mutation for an Indian population. One thing. Second, we have high percentage of mutation in non-BRCA inherited genes, as well as we have a mutation which are involved in the chemotoxicity, drug toxicity for a particular drug. So that will help the clinician to do the test before go for the treatment. Then they will have a better way to design the strategy. So you'll have a disease-free survival, better disease-free survival.